Today we're going to start Lecture 3 of Unit 4, where we're going to discuss protein and amino acid metabolism. Before we get into the metabolism of proteins and amino acids, we're first going to discuss some of the protein quality and define protein quality and discuss some of the methods that are used to evaluate the quality of protein. So protein quality is actually dependent on three different attributes. The first one is what we refer to as protein digestibility. So digestibility is actually the portion of the amino acids or the portion of the protein that can actually be digested by the animal. And this can be split. We have two different types of digestibility. One type is referred to as apparent digestibility, and that is where we have our intake minus our fecal divided by our intake. So that's apparent. So this usually underestimates our digestibility. So why does it underestimate? Well, that's where we get into what we refer to as our true digestibility. Our true digestibility is our intake minus our fecal plus our endogenous losses divided by our intake. So what are these endogenous losses? So our endogenous losses can be our any of the protein, the enzymes that are released from the pancreas, any secretions that are put into the lumen, all our mucus, all of that is our endogenous losses. In addition, we have our sloughed off cells. And then also what we can put in our endogenous losses is our microbes. So any of our microbes that die, they're going to be digested by those enzymes just like any other food source. So that's all our endogenous. And because of this, we have it that our true digestibility, by accounting for these endogenous losses, our true digestibility usually has a greater value than our apparent digestibility. The next category or attribute is our availability. Our availability a lot of times is used synonymously with digestibility, but it is a different attribute. So our availability is the actual chemical integrity of the amino acid. So if we have the Maillard reaction, for instance, so the Maillard reaction is a browning reaction. If you ever make pie, or bread, the outside of the bread or the pie is usually caused by the Maillard reaction. So it is the interaction with the amino acid, specifically lysine. It reacts with the reducing sugar. It reacts with the reducing sugar when we have oxygen and heat available. So that causes the Maillard reaction. And that makes the lysine unavailable. However, if you look at the amino acid composition, it looks like the lysine is less digestible. So digestibility and availability go hand in hand. In addition, we have problems with availability if we overcook, so our severe heat damage. So if we cook our soybean meal too much and we have a lot of protein denaturation, that's going to decrease the availability of the amino acids within that protein source. Our third attribute is our amino acid contribution or composition. So this is looking at how closely the amino acid conforms to the amino acid requirement of the animal. And this ends up resulting in what we refer to as limiting amino acids. And I'll talk about this a little bit later on in the lecture. But our limiting amino acid is the amino acid that is most deficient relative to the animal's requirement. The most common are going to be lysine, methionine, and threonine, and that's mainly in our poultry and swine. The diets that we are making, we're typically adding lysine, methionine, and threonine in the synthetic forms. So what are the methods that we utilize to evaluate our protein quality? Well, I have 4 listed here. The first one is biological value, and this is used pretty commonly, and it is evaluating the amount of protein nitrogen retained for growth and maintenance, and it's expressed as a % nitrogen digested and absorbed. So here we have our biological value, and we have our nitrogen intake. So the I equals intake. We have our nitrogen from our urine, U equals urine. Our nitrogen EU is our endogenous urine, so our E is endogenous. We have our nitrogen fecal and our nitrogen endogenous fecal. So our biological value, or BV, equals our nitrogen intake minus our urinary nitrogen minus our endogenous urine subtracted from our fecal nitrogen, accounting for our endogenous fecal nitrogen, divided by our nitrogen intake minus our fecal and our endogenous fecal. So you could see here that the biological value is accounting for what is metabolized. Our next method is our net protein utilization, or NPU, and this measures the net protein nitrogen retained as a % of the total nitrogen consumed. So it's pretty similar to our biological value. They both, our NPU and our biological value, are both measuring nitrogen. So we have our % nitrogen for our biological value and our net protein retained. The difference is it is not accounting for metabolism. So we need to be able to account for what is actually metabolized. So that's why our biological value is better than utilizing our net protein nitrogen. The next one is our amino acid scoring. And our amino acid scoring compares the protein of a feed, the amino acid profile of a feed, to a high quality reference protein. So this is widely used currently. The score is not additive unless the limiting amino acids are the same. And typically these high quality reference proteins are going to be our egg or our whey protein. Now, the downfall to our amino acid scoring, it does not look at digestibility or availability. So you can have a particular protein source that looks like it's going to be amazing, but it's just not simply digestible to the animal. So that's the limitation of the amino acid scoring. Finally, our last category is our protein efficiency ratio, and this is an old method where it is looking at the gain in body mass divided by the protein intake. Now, looking at this, I'm sure you could see many limitations of using the protein efficiency ratio. So one of the main downfalls to our protein efficiency ratio is it is not accounting for other nutrients. So if we are deficient in some of our other B vitamins, for instance, if we're deficient in our B vitamins, then the animals are not going to grow very well. They're going to have depressed growth. So we don't necessarily know if that protein is, if the protein is bad or if it is just deficient in some B vitamins. In addition, it's looking only at gain in body mass. So this does not work well if you have mature animals. So it does not work for mature animals because it's only looking at growth. In addition, it's not taking into account maintenance. So here we have our table of some of the more common proteins that are used in animal diets. So here we have our whey protein, casein, egg, and soy. So our reference proteins that I was referring to before is our whey protein and our egg. And you could see, looking at all these methods, the why. So they have the highest for our protein efficiency ratio, our biological value, our NPU, and our amino acid score. So this is why those are our reference proteins. You could see, compared to a plant protein source, that our soy is the lowest. So our plant proteins have lower protein quality than our animal proteins. So here we have a chart of some of our feed ingredients and the biological value associated with them. So you can see here at the top we have one of our reference proteins, egg albumin, which is basically right at 100. And we have our chicken, our meat meal, meat and bone meal are all pretty high. Anything under this line is plant protein. Anything above the line is an animal protein. So our animal proteins have greater biological values than our plant proteins. So you may be asking yourself, why is that? Well, one of the things, in order to get to all the wonderful protein and amino acids that the plant has available, we have to get through the cell wall. And what is that cell wall made out of? That cell wall is made out of cellulose. And if it's a more mature plant, it's going to have a significant amount of lignin associated with that. And as you remember, lignin, even bacteria can't break that apart. And our cellulose, our bacteria can break it up. And it all depends if it's a foregut or hindgut fermenter on if we're going to be able to utilize that or free that protein and get through that cell wall. So our animal protein has a greater biological value than our plant protein, and that's mainly due to the cell wall component, which is made up of cellulose and lignin.
So next we're going to discuss how we quantify and classify nutritionally our various proteins and amino acids. So what methods, what assays are done to determine our amino acid composition and protein composition. Our classic method for determining protein quantification is referred to as a Kjeldahl method. So this was developed over 100 years ago, and it is very good for determining the nitrogen content of materials. And there are 3 steps with conducting the Kjeldahl method. So first we have digestion. So this is where we cook the test substance with sulfuric acid, and that ends up forming ammonium sulfite. And so sulfuric acid is a very strong acid, and you can tell that this is not the safest assay to conduct. And typically you know that your digestion is done because the solution becomes a very, very bright, almost Kelly green. So once it gets that bright color, then you know that it's done digesting. And after that, you put it on the distillers. So during the distillation process, we are adding a very strong base, so sodium hydroxide. And that's so we can convert the ammonium that was formed with the sulfuric acid to ammonia. And then finally, we titrate that out. So we slowly add an acid back in to be able to quantitate the amount of ammonia. So once we get the amount of ammonia, we can then take that amount and multiply it by 6.25. And this is giving us our crude protein amount. So it's crude protein.
So where do we get this 6.25 correction factor when we conduct the Kjeldahl method? Well, that's coming from because the average protein contains 16% nitrogen. So if you take 100 divided by 16, it gives you 6.25. Now, one of the downfalls to this is that the proteins are going to have different amounts of nitrogen. So some of them may have amino acids that have higher nitrogen amounts than others. So this correction factor is not always accurate, but it's the one that does the best. So our Kjeldahl method is great at measuring nitrogen, and we have this limitation when we're translating that to the amount of protein. So that's why we refer to it as crude protein. So be able to tell me the amount of nitrogen that protein has and what this correction factor is. So as I mentioned before, back in 2007, we had the issue of the pet food scandal where the protein coming from China was compromised and provided melamine. So here on the right-hand side, you see the picture of melamine, and you can see all these nitrogens. So melamine is 66% nitrogen. So you can see with such a high percentage of nitrogen how that can increase the amount of, or give the false indication of a good protein quality. And when this melamine was provided in the diets with chronic ingestion, so where we have about 3% on a weight-to-weight volume, it causes toxic effects, and it affects reproduction, and more critically is it affects the kidneys in dogs. So basically the melamine was precipitating out in the kidneys, and it was causing kidney failure. So this happened in 2007, and many animals, many pets died. In addition, it was followed up, and melamine was found in baby formula in 2008. So these individuals or other companies from China were putting melamine in the diets or in the formula for infants, and many infants, a few of them died, and many of them got very critically ill. So because of this, there are a lot of companies now that really do not like to look or use protein from Chinese sources anymore. And it's also caused many companies to start looking at the actual amino acid composition as opposed to utilizing crude protein. So as I mentioned before, our limiting amino acids, these are amino acids that are found in the shortest supply relative to the amount needed for protein synthesis. And the most common ones that we are dealing with include lysine, methionine, cysteine, threonine, and tryptophan. Now, when we are discussing animals, we're most concerned about lysine, methionine, and threonine. So those are the ones that we are most commonly adding as synthetic amino acids to better meet the animal's requirement. In humans, we also have cysteine and tryptophan are often limiting amino acids for human diets. So because of these limiting amino acids, we get what we refer to as complementary proteins, to where the amino acid composition of one feed ingredient combined with the amino acids from another ends up resulting in the limiting amino acids in one cancel out the limiting amino acids in the other. So it's the complementary proteins when the rate limiting amino acids differ from the ingredients, and those complement each other. And when these two protein sources are eaten together, the amino acid from one compensates for the deficiencies in the other. So when you put these two ingredients together, it results in a higher biological value than either one ingredient on its own. So here we have our corn and our legumes are good examples. So this is what the majority of, especially poultry and swine diets are based off of, that our corn and soybeans are complementary. So you can see here that our corn has adequate methionine, valine, threonine, phenylalanine, leucine, and isoleucine, and it's deficient in tryptophan and lysine. However, when we look at what the soybeans have, they have adequate lysine, tryptophan, isoleucine, leucine, phenylalanine, threonine, and valine, and they're deficient in methionine. So when we add these two together, we end up filling in the gaps, resulting in a high quality feed. So some of our proteins are what we refer to as complete proteins. So this would be the incredible edible egg and our whey protein, our complete proteins, where they contain all the essential amino acids proportional to that which the animal requires. So our egg albumin is the standard which many of our other proteins are compared, and that's very much true for swine and poultry. Now, when you're referring to humans, they actually prefer to use whey protein as the reference because there are so many differences, or it's thought that there are differences between what a embryo chick needs versus what an adult human needs. So that's where whey protein is used as the reference for humans, but both egg albumin, which is the white, and the whey protein are both our reference proteins that we compare many of our other ingredients to, and that's because they are what we refer to as complete proteins. All right, now that we've discussed the protein quality and methods and many of those definitions, we're going to start getting into amino acid metabolism. So when we metabolize our amino acids, what are we doing with them? So some of our amino acids are being metabolized and they're actually being used for protein synthesis. So we are synthesizing more protein, and we're constantly synthesizing protein, even when you are a fully grown adult. In addition, we need amino acids to synthesize other non-essential amino acids. So we have some that are provided in the diet, others that we're able to break down and change to where we can make them our non-essential amino acids. In addition, we utilize some of our amino acids to synthesize various hormones, vitamins, pigments, and neurotransmitters. Some examples that I want you to be able to tell me include tyrosine. So tyrosine, which is a non-essential amino acid, it is able to be converted to norepinephrine. As you remember, that's a neurotransmitter associated with our sympathetic nervous system. We have epinephrine, which is also referred to as adrenaline, and that's a hormone. It is a pigment called melanin. Our thyroxine, which is controlling our basal metabolic rate, which is a hormone, and dopamine, which is another neurotransmitter. In addition, we have tryptophan, which is another good example. It's able to be converted to serotonin, which is a neurotransmitter, and it also is converted to synthesize niacin. So niacin is a B vitamin, and that's what is making up our NAD and NADP. So also with our amino acid metabolism, we metabolize amino acids during gluconeogenesis, where we're converting amino acids to glucose. They are involved in fatty acid synthesis. We utilize them for energy. They help regulate our osmotic pressure within tissues and body fluids. And then when we metabolize them, we also get various end products, including ammonia, uric acid, and urea production. So what is gluconeogenesis? And we touched on this a little bit when we were discussing carbohydrates. So gluconeogenesis is a mechanism that we use to make glucose in the liver and some species in kidneys, and we are utilizing amino acids. So our body is able to synthesize glucose from amino acids during fasting or induced glucose shortages. So these induced glucose shortages include strenuous exercise. So if you're going to go run a marathon, you are going to be breaking down your muscle protein and using those amino acids to make glucose. In addition, if you have low carbohydrate diets, so those would be the Atkins diet. There are some animals that are always gluconeogenic, so that includes our ruminants and our obligate carnivores. Our cat is also on a low carbohydrate diet, so they are always in a gluconeogenic state where they are utilizing amino acids to synthesize glucose. And we have some common starting materials, and that includes lactate or lactic acid and our glucogenic amino acids. So be able to tell me this is not leucine or lysine. So leucine and lysine are the only ones that are ketogenic strictly. They cannot be used at all to synthesize glucose. So on the exam, I will ask you that. And then we are not able to utilize fatty acids for gluconeogenesis, but we do utilize the glycerol backbone of the triglycerides. So our glycerol is also used to synthesize glucose. So this is a list of all of our various intermediates and the amino acids that can break them down, or when they are metabolized, when these amino acids are metabolized, they get these intermediate products. So we have on the top part of this red line, these are our glucogenic. So all of these are able to form glucose, and then our ones on the bottom are our ketogenic. So these are forming acetyl-CoA and acetoacetate. And you can see here that our leucine is able to form acetyl-CoA and acetoacetate. And our acetoacetate is a ketone body. Same with our lysine makes acetoacetate, which is a ketone body. And these are not able to, the acetyl-CoA and the acetoacetate are not able to go into the TCA cycle. So on the right-hand side, we have a flow diagram comparing glycolysis to gluconeogenesis, and you can tell for the most part gluconeogenesis is the reversal of glycolysis. There are some things down here at the bottom. So our pyruvate needs to be converted to oxaloacetate, and oxaloacetate is converted to phosphoenol pyruvate. So we have this added little step here, but outside of that, it is basically they use all the same enzymes once we have the phosphoenol pyruvate, and it's able to go all the way back to glucose. And gluconeogenesis takes place primarily in the liver, and in some species it occurs in the kidneys. Usually that happens when we have severe hypoglycemia, so the kidneys are able to start synthesizing glucose when we have really low blood sugar. And another reminder that our gluconeogenesis occurs from any of the amino acids except leucine and lysine. And we also have lactate, glycerol, and propionate. All of those can be utilized to synthesize glucose. So, as I mentioned, the different species have gluconeogenesis occur in different organs. So, our humans, you and I, we are able to do gluconeogenesis primarily in our liver, secondarily in our kidney. Our muscles can do a little bit, but it's primarily the liver. The liver is the main site, and that really goes for most mammals, the liver, and secondarily the kidney. The toad, interestingly, they do gluconeogenesis in the stomach. The frog is in the retina of the eye. Our fish is in the muscles, and our amphibians are also in the muscles. So gluconeogenesis is occurring within the liver, but we also utilize muscle tissue. So during our strenuous exercise, we are releasing proteins. If you're sick, you're in a gluconeogenic state, and you're breaking down proteins from your muscles. So how is your body doing that? That's what we'll be discussing over these next few slides. So your body utilizes what we refer to as the Cori cycle. So our gluconeogenesis does not directly occur in the muscle. And so we have muscle is contributing indirectly. So we have our glucose comes in, or we're breaking down our glycogen. Glycogen within the muscle is not able to enter the blood, so we break down our glucose or glycogen, and we end up getting the formation of lactate or lactic acid. So if you think of when you're exercising and you feel the burn, if you ever do aerobic videos, they'll tell you to feel the burn. Well, that is the release of lactic acid. So that is what you're feeling. That lactic acid is then able to enter the blood, and it's moved over and picked up by the liver, and your liver then converts that lactic acid back to glucose. And so it requires some ATP to have that happen, and GTP, but then we're able to take that glucose and have it go back out into the blood and be utilized by the muscles. Now, we always have protein turnover happening. We are at a constant state of synthesis and degradation. And we recycle a large portion of these amino acids during this protein turnover. And basically, we need to utilize a method for marking the protein that is no longer functioning adequately. So we utilize the ubiquitin ligase system. So ubiquitin is a small protein that's about 76 amino acids long, and it marks the old protein, and it covalently attaches to the protein to be degraded via the E3 ubiquitin ligase system, and we'll talk about that. You'll see that in the next slide. And then once we have a significant amount of ubiquitin markers on these old proteins, we have the proteasome complexes, 26S proteasome, that's able to attach to the ubiquitin, and then it degrades the protein into short peptides. And one of the first things it does is it breaks off that ubiquitin, and that ubiquitin is recycled then. So here we have a picture of ubiquitin and proteasome action, and I will say you could just disregard all this stuff in the upper right. You do not need to know any of that. unless you are really into this research. But this picture is pretty easy to follow. So we have our ubiquitin is bound to the E1, and then we have it then able to bind with the E2 and E3. Our target protein is put with the E3, and then we get this chain of ubiquitins that are put on. And this requires some energy. And we have our 26S proteasome, which is actually made up of a 20S and a 19S, and that makes our 26S. And that is able to pick up, and it detects this chain of ubiquitin on our target protein, which is marking it. So our protein goes through here. It breaks up the protein, so we get many peptides over here, and then this ubiquitin is then able to be recycled. Now, when we're discussing amino acid metabolism, we have to account for nitrogen balance. So nitrogen balance is the amount of nitrogen consumed as compared to the amount of nitrogen that's excreted in any given period of time. Now, basically, it's the total nitrogen intake minus the total nitrogen excreted, and this is excreted in the urine and the feces. Now, if you are a healthy adult, you should be at maintenance nitrogen balance, where our nitrogen intake equals the amount of nitrogen that's excreted. So protein synthesis equals protein degradation. Now, if you are a bodybuilder, if you are pregnant, if you are growing, you're going to have a positive nitrogen balance. So this is where our nitrogen intake is greater than our nitrogen output. So our protein synthesis exceeds our protein degradation. So if you're growing and you're putting on muscle tissue, if you're a bodybuilder and you're trying to build muscle mass, or if you're pregnant, where you are growing another human being inside of you, if you are lactating, where you are providing nutrition to the offspring. Those are all going to result in a positive nitrogen balance. A negative nitrogen balance is where we have our nitrogen intake is less than our nitrogen output. So this is our protein degradation exceeds our protein synthesis. And this usually occurs when you are severely ill, or if you're in a starvation state, and this is extended starvation state. So if you think of when you are ill, you do not want to eat, so therefore your body starts breaking down protein to maintain your glucose level. So it's undergoing gluconeogenesis, and you're excreting a lot more of the nitrogen in your urine. Similar things are happening during a starvation state. Never do you want to be in a negative nitrogen balance, because that is mainly indicating that you are ill or malnourished. So these next few slides, we're going to be discussing nitrogen excretion and recycling. So one of the ways specifically that our muscles, when they start degrading some muscle protein, they don't want to have ammonia enter the blood. That's going to alter our pH. So we need to be able to transfer that ammonia in the blood without altering the pH. So consequently, the muscle tissue ends up converting the amino acids to alanine. So alanine is then put into the blood, and the liver is able then to convert alanine back to glucose. So it's a way in which our muscles are able to get rid of the toxic ammonia to where our liver is able to deal with it, because only our liver is the only place where we're having the urea cycle occur. So our muscles are unable to deal with any ammonia. So we need to transfer that ammonia in the form of alanine to the liver, and then the liver is then able to convert the alanine through the it's able to convert that alanine to urea, and then that urea can then enter the blood and go to the kidneys and be excreted. Now, if you remember back from Unit 1, not all animals utilize the urea cycle to excrete nitrogen. So all mammals use the urea cycle. So any nitrogen from amino acid metabolism is used to make urea, and the urea is excreted in the urine. Now, this ends up resulting in, it requires less water than ammonia. So the urea is less toxic, so we don't need as much water. Now, our avians and our reptiles, they use nitrogen and they make uric acid. And uric acid is insoluble in water and it's excreted as a solid. So if you ever look at bird droppings or reptile droppings, you'll notice that there's a white component on the top of the droppings, and that is the uric acid. And it's insoluble in water, and because it's insoluble, it requires 50 times less water than ammonia alone. Now, fish have it the easiest because they are able to secrete nitrogen as ammonia. So as they make it, they just secrete the ammonia straight into the water, and the water that's surrounding them dilutes it out, so it is not toxic. And it's excreted from both the kidneys and the gills. And you can see here on the right-hand side, we have how these various animal groups are dealing with our ammonia. So we have ammonia here. Fish release it as ammonia. And you can see here that you need a lot more water, but it really doesn't require any energy to get rid of the ammonia. Now, as mammals, we utilize the urea cycle, which requires some energy and a little bit of water to deal with. So less water. So we're compromising. We need less water, but we require energy to deal with it. Now, our avian species and our reptiles, they make uric acid, and the synthesis of uric acid has 15 different reactions within it. So it is very costly, but it also requires less water. So those animals evolved. So many of our reptiles are living in the desert where there's inadequate water. So we wanted to make sure that they evolved to where they didn't need water to excrete and deal with the ammonia. Now, our avian species, they evolved to fly, so they needed to be able to have something lightweight. So that's why they evolved to synthesize uric acid, because if they were to make urea, it would make them too heavy and they would no longer be able to fly. So here's our urea cycle again. So you could see that we have our nitrogen, our ammonia come in, and this is occurring within the mitochondria. And it eventually binds with ornithine and is converted to citrulline. Our citrulline leaves the mitochondria. Eventually, it's converted to arginine. And then it's our arginine going to ornithine, know this part specifically, where we're getting the formation of our urea. So I'm going to expect you to be able to tell me where urea is coming off in the cycle, and is it occurring in the mitochondria or the cytoplasm? So know this cycle very well. In addition, you can see that we have some ATP that are required for the urea cycle. So the urea cycles all fine and dandy for mammals, but what about our avians and reptiles? As I mentioned before, they're utilizing uric acid. So here we have a picture on the right-hand side of the structure of uric acid, and you can see where the different components are coming from. So we have some coming from glycine, some components coming from aspartate, and then we're having some come from glutamine. So this is one of the reasons why poultry have conditionally essential amino acids related to, are they involved in the synthesis of uric acid. And on the left-hand side, you can see a picture of uric acid, and you can see that it has a very smooth texture. So one of the reasons why it has a smooth texture is that it reduces the irritation as it's going through the urogenital tract when it's being eliminated. And if you've ever, it's the same uric acid that causes gout. So if you've ever had gout, I'm sure you are well aware of how painful it is, and it is an acid, so it will burn. So we needed to, these animals evolved so it's very smooth, and if we don't get rid of it right away, we will end up having other issues with the uric acid. So having it smooth as opposed to rough helps with the irritation as it's being eliminated. So our nitrogen is recycled. So 75% of urea is excreted via the kidneys, and 25% is excreted in the colon. So we have 25% from the liver. Our urea is formed in the liver, and we have that go into the blood. 75% here is going into the kidneys. Another 25% is being brought to the colon. Now, what is happening with the urea that's being brought to the colon? So we have some of that is being broke down by bacterial urease. So bacteria are breaking down the urea, and then that is now providing a nitrogen source for them so they can synthesize their own amino acids. And those amino acids are then formed into microbial protein. And then we have some of that urea or ammonia is then being absorbed and going back into the circulatory system. So we have the urea is being broken down to ammonia again, a little bit as ammonium, and then that's entering the circulation, and then we're able to resynthesize urea. So we have this nitrogen recycling. So I want you to be able to tell me that 75% is excreted in the kidneys, 25% is occurring in the colon, and how this 25% in the colon is being dealt with. So that's basically all we're going to focus on right now related to the actual metabolism of amino acids. Now we're going to look at what happens when we have malnutrition specifically for protein. So when we have chronic protein and energy malnutrition, we get a disease that's formed called marasmus. So here you can see this child on the right-hand side that is plagued with marasmus. So this is due to both protein and energy malnutrition. So these individuals just are not getting enough to eat. It's not like they have plenty of energy and no protein, but it's both protein and energy they are insufficient in. And because of that, these children are typically short for their age. They have muscle wasting, so you can see this young child here. You can see its ribs. There's very little muscle tone at all. In addition, because these children are basically in a starvation state, they have impaired brain development since they have inadequate energy. They are using all this protein to make glucose, and the leucine and lysine that are being utilized, those are forming ketone bodies. So their brain development is impaired. And because of this, they don't have adequate energy. They don't have the heat increment, so that ends up resulting in lower body temperature. So if you remember back to our Unit 2 when we were talking about energy, some components, one of the main reasons for eating is it's help maintaining our body temperature. You have that heat increment that is being utilized to keep our body temperatures up. Well, if you have an individual that has inadequate protein and energy, they're unable to maintain that higher body temperature, so these individuals have lower body temperature. In addition, they end up having many issues with the development of the epithelium within their GI tract, which results in nutrient malabsorption. So even if they have adequate vitamins and minerals within their diet, they are not necessarily able to absorb those very efficiently because they are lacking the protein and energy to maintain the proper GI tract. The next disease state is referred to as kwashiorkor, and that is an acute protein malnutrition where marasmus is chronic in its protein and energy. Kwashiorkor is basically because it's strictly because of protein, and kwashiorkor actually is Gonin, meaning that it is an evil spirit that infects the first child when the second is born. So typically in many of these countries that have inadequate resources to good food, they will breastfeed their children for 2 to 3 years. And that they usually will end up stopping when they get pregnant again. So once they are pregnant with their second child, they now need to, that breast milk needs to be devoted to the second child, and the older one is usually more self-sufficient. So this occurs very rapidly upon weaning. So usually these children are moved from the milk protein, which is very nutritious and it's meeting their nutritional requirements, to upon weaning, they're usually given a more poor quality protein source. So, and usually it's a single source, so usually it could be rice. You can have beans, although beans are good proteins. They're not as high quality as the animal protein. So it results in acute protein malnutrition. So you could see here we have a child both before when it had kwashiorkor, and you can see here that the abdominal is pretty swollen. They do have some muscle wasting. Upon, if any of them die, you could see they have fatty liver, and they usually are also plagued with many skin lesions. And once they are provided adequate protein, they are able to bounce back rather quickly. So you can see this is the same child. They no longer has a swollen abdomen, and they are able to come back. So next we're going to talk about some of the aberrations involved in protein metabolism. One of the main aberrations is associated with the prions. So prions are proteinaceous particles that do not contain nucleic acids, and they are mostly an abnormal, consist mostly of an abnormal form of prion protein. So our prion proteins are found naturally. So our normal prions are PRPC, and they're normally found within the membrane in the brain and the neural tissue. And then these prions can become abnormal. So our abnormal prions become PRPSC, and these when we have a PRPSC, that will actually cause normal prions to alter their shape, and they then become abnormal prions. And when these rogue abnormal prions infect an animal, it causes the host encoded stimulatory RNA to convert the normal prions to these rogue prions. And then these rogue prions are causing various degenerative brain diseases in which there is no cure. So the main diseases caused by these rogue prions are the transmissible spongiform encephalopathy, or TSE. So we have, depending on the species that's infected, we have different terms. So the most common that you've probably heard about is bovine spongiform encephalopathy, or BSE. It's also referred to as mad cow disease. And there was outbreaks back in the mid-90s in Great Britain, where a population was many individuals, so ended up developing Crutchfield-Jacobs disease, which is the human version of TSE, and it was concluded that those individuals developed the disease because there were cattle that were found to have BSE. So now, because of that, the USDA utilizes a great number of resources to where they can monitor this, and we've never had any cattle in the U.S. come down with mad cow. A few years ago there was one, but it actually came from Canada. It was not raised in the U.S., and they had it tracked. But many regulations have gone into the USDA related to downer cows, so you're no longer allowed to put any sick animals and harvest those. So those have to just be euthanized at the farm. They can't be put into the food chain. In addition, you cannot consume any neural tissue at all from any animal species because of the threat of the TSE. In sheep, it's referred to as scrapie. And then we have deer and elk, so it does occur naturally. That's referred to as chronic wasting disease. As I mentioned, the Crutchfield-Jacobs disease is in humans. There also is Kuru, which that is mainly associated with cannibalism. So as long as you don't practice cannibalism, you don't have to worry about that one. So why are these prions, these rogue prions, so scary? Well, one of the reasons is they are resistant to physical and chemical treatments. So you can't really reverse them, you can't stop them. They're resistant to many enzymes except keratinase. There are some keratinases that are able to break down these prions, but none of the other enzymes. So if you consume it, your pancreatic proteases are unable to break down these prions. In addition, you don't have any immune or inflammatory response, and because the body is not detecting these as non-self or harmful molecules, then we're unable to mount an immune response. So if our innate immune system does not recognize these prions as foreign, it's not going to mount any sort of T cell or B cell response. In addition, it has a very wide range of pH tolerance. So it can handle a pH down to 2.5, and it can handle it all the way up to 10.5 before they become denatured. So many of these prions are able to survive the very harsh pH that they're exposed to within the stomach. So what's the difference between our normal prions and these PRPSE rogue prions? Well, a lot of that has to do with the secondary structure, as we mentioned in the first lecture of this unit on the different structures. So our prions have both alpha helices and beta pleated sheets. However, you can see that they have, our normal ones have about 43% alpha helices. And these rogue prions actually end up increasing the amount of our beta pleated sheets. So then this increases to 43%, resulting in 30% as alpha helices. So this is the main difference, is the secondary structure is causing these rogue prions. So it's basically a misfolding of the protein. So what happens to the brain tissue when you have these rogue prions? Well, basically you get excessive vacuolization. So we have many holes that end up developing within the brain, and we have dark plaques. So you can see all these little white circles in here. So that's a lot of the vacuolization. In addition, we have these black spots. So we have increased plaque in all versions. So here we have BSE, which is cattle. We have Crutchfield-Jacobs disease, which is human, and Scrapie, which is sheep. Now, if you look and you do a PET scan on individuals, normal individuals, and one that has Crutchfield-Jacobs disease, you could see that the metabolism of glucose is altered. So our glucose metabolism is reduced in individuals that have Crutchfield-Jacobs disease. It makes sense because we have more vacuolization, so we have less cells that are surviving. So therefore, we have decreased glucose metabolism. So how do these prions enter? So obviously, we're unable to break them down with these enzymes. But you would think, okay, our epithelial cells have these nice tight junctions. That's the whole reason we keep bacteria out. So how are these prions able to be absorbed? Basically, there's these cells within the GI tract referred to as the M cells. And these cells are there, and they are basically, they allow some bacteria components to come through and meet the macrophages and other immune cells underneath them. So it's a way that your immune system can be checking what's within the lumen, what's good or bad. So these prions are able to, and these M cells are pretty porous, so they're intentionally porous so we can have this bacteria come through. However, these M cells, which make up about 1% of the epithelium, they allow them to come through, and then they're not taken up by our macrophages because there's no way for our macrophages to identify that these are non-self. So then these prions are able to eventually make it to the blood, and they can get into the brain tissue. All right, our last slide is on sickle cell anemia. So this is the last aberration that we're going to be discussing. So sickle cell anemia is a genetically induced aberration in protein folding, and basically it's our red blood cells end up carrying less oxygen because they have this irregular sickle shape. So here on this bottom right, you can see we have our normal red blood cell, a nice circle, and then right underneath it we have a sickle-shaped red blood cell. So this sickle shape ends up clogging the capillaries. So if you remember from your physiology, our capillaries are small blood vessels that can basically only carry one blood cell at a time. So the sickle shape ends up sticking to vessel walls, and it clogs it up, so we have reduced flow, and consequently we have reduced tissue oxygenation. So our sickle cells, red blood cells they're not able to carry as much oxygen. In addition, they're clogging these capillaries, which is reducing the amount of normal red blood cells from going through. And we have many ethnicities that are prone to sickle cell anemia. Many people are very well aware that African Americans are very susceptible. And then we have other Middle Eastern countries, including Arabia, Greece, Italy. All of those ethnicities are susceptible to sickle cell anemia. Latin Americans, and then Indians are also very susceptible to sickle cell anemia. The most common one is going to be African American individuals. So we have 1 in 400 African Americans that are at risk of having sickle cell anemia. So this is all due to that misfolding of protein that we talked about in the last lecture.